What is the RNA Tie Club?
In 1953, shortly after the publication of James Watson and Francis Crick's paper on the structure of DNA, George Gamow, 1904 to 1968, a Russian physicist, wrote to Watson and Crick and suggested a mathematical link between DNA structure and the structure of 20 amino acids. The RNA Tie Club was an outgrowth of this proposal. Members included Watson, Crick, and 17 others, each taking a nickname from one of the 20 amino acids. Each member of the club was presented with a specially designed tie corresponding to the appropriate amino acid.
What are Koch's postulates?
Koch's postulates are the 4 basic criteria an organism must meet in order to be identified as pathogenic, capable of causing disease. The characteristics are as follows: 1. The organism must be found in tissues of animals that have been infected with the disease, rather than in disease-free animals. 2. The organism must be isolated from the diseased animal and grown in a pure culture or in vitro. 3. The cultured organism must be able to be transferred to a healthy animal, which will show signs of the disease after having been exposed to the organism. 4. The organism must be able to be isolated from the infected animal.
Why will my dog play fetch but not my cat?
Domestication occurred differently for cats and dogs. While dogs came to work closely with humans, relying on them for food and shelter, cats maintained a more distant relationship. A dog plays fetch because it is an extension of the cooperative working hunting relationship she has with humans, while the cat has no such relationship.
What is scanning tunneling microscopy?
Scanning tunneling microscopy, STM, also called a scanning probe microscopy, was developed in the 1980s to explore the surface structure of specimens at the atomic level. This technique uses electronic methods to move a metallic tip, a conducting material such as platinum iridium, composed ideally of a single atom, across the surface of a specimen. As the tip is moved across the surface of the specimen, electrical voltage is applied to the surface. If the tip is close enough to the surface and the surface is electrically conductive, electrons will begin to leak or tunnel across the gap between the probe and the sample. The tip of the probe is automatically moved up and down to maintain a constant rate of electron tunneling across the gap as the probe scans the sample. The movement is presented on a video screen. Successive scans then build up an image of the surface at atomic resolution.
How does a nematocyst work?
A nematocyst is a specialized organelle found in all cnidarians. Each nematocyst features a coiled, thread-like tube lined with a series of barbed spines. The nematocyst is used to capture prey and may also be used for defense purposes. When it is triggered to discharge, the extremely high osmotic pressure within the nematocyst, 140 atmospheres, causes water to rush into the capsule, increasing the hydrostatic pressure and expelling the thread with great force. The barb instantly penetrates the prey, stinging it with a highly toxic protein.
What are the commercial uses of brown algae?
Kelp, one of the species of brown algae, is harvested from ocean water for the sodium, potassium salts, iodine, and alginates that it contains. The alginates are carbohydrates used in the formation of gels.
What naturally occurring substance provides protection against viral infections?
Interferons protect the adjacent cells against viral penetration. Interferons are glycoproteins produced by body cells upon exposure to a virus. In 1957, Alec Isaacs, 1921 to 1967, and Jean Lindenmann, 1924, identified a group of over 20 substances that were later designated as alpha, beta, and gamma interferons.
When was the first plant patent issued?
Henry F. Bosenberg, a landscape gardener, received U.S. plant patent number 1 on August 18, 1931. For a climbing or trailing rose.
What is an operon?
An operon is a segment of DNA containing all genes used to produce proteins in a specific metabolic pathway. A functional operon also contains the RNA polymerase binding site, known as a promoter, as well as an on-off switch known as the operator. So far, operons have been identified only in bacteria. As this highly efficient grouping of related genes maximizes the information encoded on their single chromosome.
Do camels store water in their humps?
The hump or humps do not store water, since they are fat reservoirs. The ability to go long periods without drinking water, up to 10 months if there is plenty of green vegetation and dew to feed on, results from a number of physiological adaptations. One major factor is that camels can lose up to 40% of their body weight with no ill effects. A camel can also withstand a variation of its body temperature by as much as 14 degrees Fahrenheit 8 degrees Celsius. A camel can drink 30 gallons, 113.5 liters, of water in 10 minutes and up to 50 gallons, 189 liters, over several hours. A one-humped camel is called a dromedary or Arabian camel. A Bactrian camel has 2 humps and lives in the wild on the Gobi Desert. Today, the Bactrian is confined to Asia, while most of the Arabian camels are on African soil.
How can bryophytes be used as bioindicators?
Bioindicators are physiological, chemical, or behavioral changes that occur in organisms as a result of changes in the environment. Bryophytes of the genus Hypnum are particularly sensitive to pollutants, especially sulfur dioxide. As a result, most bryophytes are not found in cities and industrial areas. Mosses and liverworts, especially Hypnum cupressiforme and Homolitecium sericium, were used as bioindicators to monitor radioactive fallout from the Chernobyl reactor accident in 1986.
What is meant by a diploid organism?
A diploid organism is one that has 2 sets of chromosomes, one from each parent.
When were mitochondria discovered?
In 1857, Rudolf Albert von Kolliker, 1817 to 1905, histologist and embryologist, first described sarcosomes, now called mitochondria, in muscle cells. The term mitochondrion, meaning thread-like granule, was first used in 1898. Functionally active mitochondria were first isolated in 1948. Kolliker was among the first biologists to interpret tissue structure in terms of cellular elements.
What is the difference between an endangered species and a threatened species?
An endangered species is one that is in danger of extinction. Throughout all or a significant portion of its range. A threatened species is one that is likely to become endangered in the foreseeable future.
In other words, how many different species are there within a habitat?
Finally, measuring ecosystem diversity is an attempt to keep track of the loss of different types of habitats. This in turn gives scientists a sense of what types of species are going extinct at any given time.
What are applications of DNA fingerprinting?
DNA fingerprinting is used to determine paternity, in forensic crime analysis. In population genetics to analyze variation within populations or ethnic groups, in conservation biology to study the genetic variability of endangered species. To test for the presence of specific pathogens in food sources. To detect genetically modified organisms either within plants or food products, in evolutionary biology to compare DNA extracts from fossils to modern-day counterparts, and in the identification of victims of a disaster.
What is transduction?
Transduction is the process by which a vector, usually a bacteriophage, carries DNA from one bacterium to another bacterium. It can be used experimentally to map bacterial genes.
Where do transcription and translation take place?
In bacteria, the process of protein synthesis takes place in the cytoplasm. As bacteria lack a nucleus or other membrane-bound organelles. In eukaryotes, transcription occurs in the nucleus. From there the mRNA moves into the cytoplasm, where it is translated into a polypeptide at a ribosome.
What is the most popular state insect?
The honeybee is by far the most popular state insect, having been selected by 16 states, Arkansas, California, nicknamed the Beehive State, Georgia, Kansas, Louisiana, Maine, Mississippi, Missouri, Nebraska, New Jersey, North Carolina, Oklahoma, South Dakota, Tennessee, Vermont, and Wisconsin.
How was it discovered that microorganisms are effective against bacterial infections?
British microbiologist Alexander Fleming, 1881 to 1955. Happened upon the discovery of penicillin's use as an antibacterial agent. In 1928 Fleming was researching staphylococci at St. Mary's Hospital in London. As part of his investigation, he had spread staphylococci on several petri dishes before going on vacation. Upon his return he noticed a green-yellow mold contaminating one of the petri dishes. The staphylococci had failed to grow near the mold. He identified the mold as being of the species Penicillium notatum. Further investigation proved that staphylococci and other gram-positive organisms are killed by P. notatum. It was not until the 1940s that Howard Florey, 1898 to 1968, and Ernst Boris Chain, 1906 to 1979, rediscovered the benefits of penicillin and were able to isolate it for medical use. In 1945 Fleming, Florey, and Chain shared the Nobel Prize in Physiology or Medicine for their work on penicillin.
What is a gene chip?
A gene chip is part of the process of microarray profiling. It is also known as a biochip or a DNA chip. It is about the size of a postage stamp and is based on a glass wafer, holding as many as 400,000 tiny cells. Each tiny cell can hold DNA from a different human gene and can perform thousands of biological reactions in a few seconds. These chips can be used by pharmaceutical companies to discover what genes are involved in various disease processes. They can also be used to type single nucleotide polymorphisms, SNPs, which are base pair differences that are found approximately every 500 to 1000 base pairs in DNA. There are more than 3 million SNPs in the human genome. They are very important in DNA typing because they represent about 98% of all DNA polymorphisms.
What is DNA?
Deoxyribonucleic acid, DNA, is the genetic material for all cellular organisms. The discovery of DNA is considered the most important molecular discovery of the 20th century.
What factors affect enzyme function?
Enzymes may be controlled at a variety of levels, since proteins are coded with DNA. A change in DNA will change the rate at which an enzyme is produced. Control molecules known as competitive inhibitors may prevent the substrate from reaching the enzyme. Allosteric or non-competitive regulators may bind elsewhere on the enzyme, causing its shape to change and thereby altering its active site. Changes in environmental conditions such as pH, temperature, or salt concentration also affect shape. Absence of required enzyme helpers such as vitamins and minerals.
When was the term dinosaur first used?
The term dinosaur was first used by Richard Owen, 1804 to 1892, in 1841 in his report on British fossil reptiles. The term, meaning fearful lizard, was used to describe the group of large, extinct reptiles whose fossil remains had been found by many collectors.
What are the functions of the root system?
The major functions of roots are, 1. Anchorage in soil. 2. Storage of energy resources such as the carrot and sugar beet. 3. absorption of water and minerals from the soil. And 4. Conduction of water and minerals to and from the shoot. The roots store the food, energy resources, of the plant. The food is either used by the roots themselves or digested. And the products of digestion are transported back up through the phloem to the above ground portions of the plant. The roots of some plants are harvested as food for human consumption. Plant hormones are synthesized in the meristematic regions of the roots and transported upward in the xylem to the aerial part of the plant to stimulate growth and development.
What is the root cap?
The root cap is a thimble-like mass of parenchyma cells that covers and protects the growing root tip as it penetrates the soil. The root cap is pushed forward as the root tip grows longer. The cells on the periphery of the root cap are sloughed as the root cap is pushed forward and new cells are added by the apical meristem. The root cap protects the apical meristem, aids the root as it penetrates the soil, and plays an important role in controlling the response of the root to gravity, gravitropism.
What is a metabolic pathway?
A metabolic pathway is a series of interconnected reactions that share common mechanisms. Each reaction is dependent on a specific precursor, a chemical, an enzyme, or the transfer of energy. One of the first studies of metabolic pathways was carried out in 1909 by the British physician Archibald Garrod, 1857 to 1936. His study suggested a link between the inability to make a particular enzyme and inherited disease. The disease was alkaptonuria, a condition in which urine darkens upon exposure to air, due to the presence of the chemical alkapton. Garrod's discovery was one of the first incidents of a physical manifestation being tied to a specific metabolic disorder.
What is translation?
Translation is the process that produces polypeptides from an mRNA transcript. This process involves several different types of molecules: tRNA, rRNA, ribosomal proteins, and the energy molecule GTP.
How much garbage does the average American generate?
According to the Environmental Protection Agency, nearly 232 million tons of municipal waste was generated in 2000. This is equivalent to 4.6 pounds 2.1 kilograms per person per day, or approximately 1,700 pounds 770 kilograms per person per year.
Why do puffer fishes puff?
Pufferfishes are any of a number of species found in warm seas that use a special adaptation of the gullet to inflate their bodies to nearly twice the normal size. Pufferfishes, blowfishes, and their like do this in response to a perceived threat. The increased size and unpalatable-looking spines make the potential prey look quite unappetizing to predators.
How is the vertebrate brain organized?
The vertebrate brain is divided into three regions: the hindbrain, the midbrain, and the forebrain. The size of each region of the brain varies from species to species. The hindbrain may be considered an extension of the spinal cord. Hence, it is often described as the most primitive portion of the brain. The primary function of the hindbrain is to coordinate motor reflexes. The midbrain is responsible for processing visual information. The forebrain is the center for processing sensory information in fish, amphibians, reptiles, birds, and mammals.
How does the term negative feedback apply to metabolism?
Negative feedback is a cellular process that is similar to the manner in which an air conditioner operates. An air conditioner can be set to a certain temperature, and when the surrounding air reaches the set temperature, the air conditioner shuts off. Negative feedback is part of the homeostatic process through which cells conserve energy by synthesizing products only for their immediate needs.
What is a molecular clock?
The molecular clock is a hypothesis that assumes that random mutation in terms of the substitution of one nucleotide for another occurs at a linear rate. By comparing the number of nucleotide substitutions between two species, scientists could then estimate the amount of time that has elapsed since they shared that sequence with their most recent common ancestor.
Why are some strains of bacteria pathogenic and other strains not?
Strains of bacteria possess genetic differences. These differences are not sufficient for them to be considered as separate species, but each strain is distinctive. For example, 98 there are many different strains of Escherichia coli, E. coli. Some, such as E. coli 0157H7, cause serious diseases, while others live in the intestine and can be considered beneficial, as they aid digestion.
What was the typical lifespan of dinosaurs?
the lifespan has been estimated at 75 to 300 years. Such estimates are educated guesses. From examination of the microstructure of dinosaur bones, scientists have inferred that dinosaurs matured slowly and probably had proportionately long lifespans.
What is centrifugation?
Centrifugation is the separation of immiscible liquids or solids from liquids by applying centrifugal force. Since the centrifugal force can be very great, it speeds the process of separating these liquids instead of relying on gravity.
What are killer bees?
Africanized honeybees. The term entomologists prefer rather than killer bees are a hybrid originating in Brazil, where African honeybees were imported in 1956. The breeders, hoping to produce a bee better suited to producing more honey in the tropics, instead found that African bees soon hybridized with and mostly displaced the familiar European honeybees. Although they produce more honey, Africanized honeybees, Apis mellifera scutellata, also are more dangerous than European bees because they attack intruders in greater numbers. Since their introduction, they have been responsible for approximately 1,000 human deaths. In addition to such safety issues, concern is growing regarding the effect of possible hybridization on the U.S. beekeeping industry. In October 1990, the bees crossed the Mexican border into the United States. They reached Arizona in 1993. In 1996, 6 years after their arrival in the United States, Africanized honeybees could be found in parts of Texas, Arizona, New Mexico, Nevada, and California. Their migration northward has slowed, partially because they are a tropical insect and cannot live in colder climates. Experts have suggested two possible ways of limiting the spread of the Africanized honeybees. The first is drone flooding, a process by which large numbers of European drones are kept in areas where commercially reared European queen bees mate, thereby ensuring that only limited mating occurs between Africanized drones and European queens. The second method is frequent requeening, in which a beekeeper replaces a colony's queen with one of his or her own choosing. The beekeeper can then be assured that the queens are European and that they have already mated with European drones.
What is meant by heterozygote advantage?
Heterozygote advantage, also known as balancing selection, arises when there is a survival selective advantage for hybrids, heterozygotes, as compared to that of either homozygous recessive genotype or homozygous dominant genotype. Some diseases are lethal only in the homozygous state.
What is cell fluorescence?
Fluorescence is luminescence caused by a pigment molecule. Pigments are molecules that absorb some colors of light while reflecting others. For example, green pigments, like those in leaves, absorb red and blue wavelengths of light but reflect green wavelengths. After being energized by photons, units of light energy, the electrons of some pigment molecules actually give off light as they fall back to their normal state. Chlorophyll molecules that play a role in photosynthesis have this ability, as do molecules found in several organisms, such as jellyfish. The body of the insect Photinus pyrolis, better known as a lightning bug, has the enzyme luciferase, which generates the chemical reaction that leads to a drop in the energy state of electrons A. Reaction similar to that which occurs in a chlorophyll molecule, allowing the insect to glow.
What is a redox reaction?
A redox reaction is actually a series of chemical reactions involving the transfer of electrons from one atom or molecule to another. By transferring electrons, the cell is also transferring energy. The molecule that loses an electron is said to be oxidized, resulting in its charge becoming positive. The molecule that gains an electron is said to be reduced, and its charge becomes negative. Electrons do not travel alone in cells, rather, they are accompanied by a proton, H+. A proton and an electron together make a hydrogen atom. Ultimately, a redox reaction involves the loss of hydrogen atoms from one molecule and the gain of hydrogen atoms by another molecule.
What is ultrasound?
Ultrasound is another type of 3D computerized imaging. Using brief pulses of ultra-high frequency acoustic waves, lasting 0.01 second, it can produce a sonar map of the imaged object. The technique is similar to the echolocation used by bats, whales, and dolphins.
How do stomach cells survive the acid in the stomach?
The cells that line your stomach produce a basic, alkaline mucus. The mucus has a higher pH that counteracts the acid produced by the stomach, which has a pH of 2.0. Because of this, cells are protected from the digestive enzymes at work in the stomach. If stomach acid manages to reach the tissue below the protective mucus layer, ulcers can result.
What is X-ray crystallography?
X-ray crystallography, also called X-ray diffraction, is used to determine crystal structures by interpreting the diffraction patterns formed when X-rays are scattered by the electrons of atoms in crystalline solids. X-rays are sent through a crystal to reveal the 534 pattern in which the molecules and atoms contained within the crystal are arranged.
How is commercial cork cultivated?
Commercial cork is the outer bark of the cork oak, Quercus suber, grown in the western Mediterranean. The first periderm is commercially useless. It is removed from the tree and discarded when the tree is approximately 10 years old. When the tree is 20 to 25 years old and has a diameter of approximately 15.75 in 40 centimeters, a usable cork layer 1.2 to 3.9 in 3 to 10 centimeters thick can be harvested. A similar layer can be harvested approximately once every 10 years until the tree is approximately 150 years old. The cork of a cork oak breaks away at the cork cambium and can be peeled off without harming the tree.
How does caffeine affect cells?
Caffeine is probably the most common drug ingested by people worldwide. Caffeine affects cells by stimulating lipid metabolism and slowing the use of glycogen as an energy source. As a whole, the body responds to caffeine by extending endurance, allowing you to stay awake for longer periods of time or perform extra activities. Adverse effects of excess caffeine intake include stomach upset, headaches, irritability, and diarrhea.
What is sexual selection?
When individuals of the same sex differ in their mating success and that difference is correlated to the presence or absence of a particular trait, that trait is said to be the result of sexual selection. Traits that provide no benefit to survival but that increase the likelihood of the male acquiring a mate are examples of sexual selection.
What methods are used to estimate wildlife populations?
Since it is usually impossible, and often impractical, to count all individuals in a population, researchers use a variety of sampling techniques to estimate population densities. One method is to count the individuals in a certain area. The larger the number and size of sample plots, the more accurate the estimates. Population densities may also be estimated based on indirect indicators such as animal droppings or tracks, nests, or burrows.
What types of samples can be used for DNA fingerprinting?
Any body fluid or tissue that contains DNA can be used for DNA fingerprinting, including hair follicles, skin, earwax, bone, urine, feces, semen, or blood. In criminal cases, DNA evidence may also be gathered from dandruff, from saliva on cigarette butts, chewing gum, or envelopes, and from skin cells on eyeglasses. What is the largest forensic DNA investigation in U.S.?
How does an ultramicrotome differ from a standard microtome?
The ultramicrotome, developed after 1950 to prepare specimens for examination under the electron microscope, enables technicians to cut very thin sections, 50 to 150. Instead of embedding specimens in wax, small, 0.5 to 1.0 cubic mm. Biological specimens are embedded in very hard synthetic resins such as epoxy. In order to cut these very hard materials, special knives sharper than steel knives are used. The knives are either diamond knives or pieces of plate. Glass broken in a controlled way to produce a fine edge.
Do animals laugh?
So far, no example of animal laughter comparable to that of humans has been documented. However, researchers have reported that under certain conditions some species emit special vocalizations that could be considered akin to laughter. In one specific case, a researcher has been able to identify a huffing sound unique to dogs at play. Scientists studying the behavior of non-human primates and rats have reported similar observations.
Why do cows have 4 stomachs?
Cows have 4 stomachs in order to process their low-quality diet of grass. The 4 sections are, 1. The rumen, 2. The reticulum, 3. The omasum, and 4. The abomasum. Cows are a type of mammal known as a ruminant. Ruminants eat rapidly and do not chew much of their food completely before they swallow it. The liquid part of their food enters the reticulum first, while the solid part of their food enters the rumen, where it softens. Bacteria in the rumen initially break the food material down as a first step in digestion. Ruminants later regurgitate the partially liquefied plant parts into the mouth, where they continue to munch it in a process known as chewing their cud. Cows chew their cud about 6 to 8 times per day, spending a total of 5 to 7 hours in rumination. The chewed cud goes directly into the other chambers of the stomach, where various microorganisms assist in further digestion. Herbivores have a longer small intestine to allow maximum time for the absorption of nutrients.
What is gene redundancy?
Gene redundancy refers to having multiple copies of the same gene. This assures that essential genes that are required in large amounts, like those for ribosomal RNA, rRNA, can be transcribed at multiple sites. In addition, gene redundancy provides for a source of gene sequences that can be modified by natural selection without immediately destroying the organism.
How do cells recognize each other?
Cells recognize each other through the molecules that are attached to their extracellular matrix, which fills the space between cells, binding cells, and tissues together.
When were bacteriophages first discovered?
Bacteriophages were discovered in the early 1900s by Frederick W. Toward, 1877 to 1950, a British scientist, and Felix D. Harrel, 1873 to 1949. A French scientist. In 1915 Toward reported observing a filterable agent that destroyed bacteria growing on solid media. D. Harrel independently confirmed the discovery in 1917. It was actually D. Harrel who named the agent bacteriophage. However, very few scientists accepted these findings and the work on the growth and infectious nature of bacteriophages. It was not until the 1930s that Martin Schlesinger, a German biochemist, characterized bacteriophages, establishing their own unique place in the microbial world.
Who was Erasmus Darwin?
Erasmus Darwin, 1731 to 1802, was the grandfather of Charles Darwin, 1809 to 1882. Erasmus was a physician, inventor, and natural scientist who published a book on his ideas. Zoonomia, between 1794 and 1796. Zoonomia contained poetic couplets describing Erasmus Darwin's ideas about science and, in particular, the evolution of life. Erasmus Darwin's hypothesis was that all the animals on the planet had their origin in a vital spark that set in motion life as we know it. In his posthumously published book Temple of Nature, 1803, Erasmus Darwin further speculated on a theory of evolution that included basic ideas on the unity of organic life and the importance of both sexual selection and the struggle for existence in the evolutionary process. Years later, in writing a biography about his grandfather, Charles Darwin acknowledged that his grandfather's ideas had influenced his thinking.
Which fungi commonly found in North America have demonstrated beneficial medicinal effects?
Clinical trials have been conducted to test potential health benefits of fungi commonly found in North America. The following chart reveals some of the benefits that several common fungi may provide.
What causes the Mexican jumping bean to move?
The bean moth, Carpacappa saltitans, lays its eggs in the flower or in the seed pod of the spurge, a bush known as Euphorbia sebastiana. The egg hatches inside the seed pod, producing a larva or caterpillar. The jumping of the bean is caused by the active shifting of weight inside the shell as the caterpillar moves. The jumps of the bean are stimulated by sunshine or by heat from the palm of the hand.
How are coral reefs formed and how fast are they built?
Coral reefs grow only in warm, shallow water. The calcium carbonate skeletons of dead corals serve as a framework, upon which layers of successively younger animals attach themselves. Such accumulations, combined with rising water levels, slowly lead to the formation of reefs that can be hundreds of meters deep and long. The coral animal, or polyp, has a columnar form. Its lower end is attached to the hard floor of the reef, while the upper end is free to extend into the water. A whole colony consists of thousands of individuals. There are two kinds of corals, hard and soft, depending on the type of skeleton secreted. The polyps of hard corals deposit around themselves a solid skeleton of calcium carbonate, chalk, so most swimmers see only the skeleton of the coral. The animal is in a cup-like formation into which it withdraws during the daytime. The major reef builder in Florida and Caribbean waters, Montas triannularis. Star coral, requires about 100 years to form a reef just 3 feet 1 meter high.
Why is there such a range of skin color in humans?
Studies on the genetic components of skin color indicate that at least 3 and perhaps as many as 4 gene pairs interact to produce a given shade of skin color. 4 gene pairs would generate 4x4x4x4, 256, possible genotypes, which is ample for the number of color variations found among humans.
What is analogy?
To an evolutionist, an analogous structure is one that looks similar or has the same purpose but is definitely not the result of common inheritance. For example, bats and birds both use wings to fly, and the wings have the same general shape, thin but broad in width. However, the structures were not inherited from the same ancestors. Bats were four-legged mammals before their front limbs became modified for flight. While birds are not descended from mammals at all.
Do possums play dead?
The common or Virginia opossum, Didelphis virginiana, is the only marsupial in the southern and eastern portions of the United States. When threatened or frightened, the opossum will lie quite still with stiffened limbs and a fixed gaze, appearing as if dead. When the perceived threat is over, the opossum will return to its normal, nocturnal activities.
How is animal behavior studied in the field?
Animal behavior is studied by construction of an ethogram, which is a listing and description of all naturally observed behaviors. Behavior can also be studied through the use of manipulative investigations, both in the field and in the laboratory. These behaviors are then categorized. In order to be objective, all observers must record behavior patterns in exactly the same way. Observations can then be statistically analyzed.
Could the events depicted in Jurassic Park actually happen?
In this novel, later adapted into a film, about dinosaur reincarnation, scientists bring dinosaurs back to life by using dinosaur DNA, obtained from insects embedded in fossilized amber. There are several flaws in this scenario. One is that prehistoric mosquitoes were most likely to have digested the dinosaur blood, making the DNA unusable. Also, one would need a complete genome, not one that has been augmented with amphibian DNA, to genetically reconstruct a dinosaur. The best way that dinosaurs could be recreated would probably be by altering bird DNA. Since dinosaurs and birds are genetically related.
What is cognition?
Cognition is the highest form of learning, and consists of the perception, storage, and processing of information gathered by sensory receptors.
What is kudzu?
Kudzu, pure Aria lobata, is a vine that was brought from Japan for the 1876 Centennial Exposition in Philadelphia. It was intentionally planted throughout the southern United States during the 1930s in an attempt to control erosion. In fact, the federal government paid farmers as much as $8 an acre to plant it. In 1997, however, the government reversed its position on kudzu and referred to it as a noxious weed. Kudzu grows over everything that it encounters, draping itself across power poles and pine trees like a shawl. The plant is responsible for more than $50 million in lost farm and timber production each year. It grows at a rate of 120,000 acres per year. As of the early 21st century, it covers between 2 and 4 million acres of land throughout the United States, occurring from Connecticut in the east to Missouri and Oklahoma in the west, and south to Florida and Texas. Kudzu grows as fast as 1 foot 30 centimeters per day. The latest approach to controlling the growth of kudzu is to have goats chew on it, devouring the leaves, stems, and roots.
What are the steps of the gram stain?
Crystal violet, a primary stain, one that imparts its color to all cells, is applied to a heat-fixed smear. After a short time, the crystal violet is washed off, and the smear is covered with iodine, a mordant. When the iodine is washed off, the bacteria appear dark violet or purple. The slide is then washed in an alcohol or alcohol acetone solution. This decolorizing agent removes the purple from the cell of some species but not from others. The alcohol is rinsed off, and the slide is then stained with safranine, a basic red dye. The smear is then washed again, blotted dry, and examined under a microscope. Gram-positive bacteria retain the purple dye, while those that lose the purple color are classified as gram-negative.
How is chromatography used to identify individual compounds?
Chromatography is another technique used to separate mixtures into their individual components. All methods of chromatography share common characteristics. The process is based on the principle that different chemical compounds will stick to a solid surface or dissolve in a film of liquid, to different degrees. Chromatography involves a sample, or sample extract, being dissolved in a mobile phase, which may be a gas, a liquid, or a supercritical fluid. The mobile phase is then forced through an immobile, immiscible stationary phase. The phases are chosen such that components of the sample have differing solubilities in each phase. The least soluble component is separated first, and as the separation process continues, the components are separated by increasing solubility.
What is myelin?
Myelin forms an insulating wrapping around large nerve axons. In the peripheral nervous system, myelin is formed by Schwann cells, a type of supporting cell, that wrap repeatedly around the axon. In the central nervous system, myelin is formed by repeated wrappings of processes of oligodendrocytes. A different type of supporting cell. The process of each cell forms part of the myelin sheath. The space between the myelin from individual Schwann cells or oligodendrocyte processes is a bare region of the axon called the node of Ranvier. Nerve conduction is faster in myelinated fibers because it jumps from one node of Ranvier to the next. For this reason it is called saltatory, jumping, conduction.
How does sickle cell anemia affect the function of red blood cells?
The genetic defect in sickle cell anemia is a mutation in one of the polypeptide chains that comprise the hemoglobin molecule. The abnormal molecule produced in sickle cell anemia causes a change in the shape of affected red blood cells. The disease morphs a typically rounded red blood cell disc into a crescent or sickle shape. The abnormal red blood cells affected by sickle cell anemia are rigid and are more likely to clump together. Because of their tendency to group, they are more prone to sticking to the walls of blood vessels. They can even block the blood vessels themselves. Because of this, the hemoglobin molecule isn't able to carry oxygen as well as the cells need it to.
What are secondary metabolites and what are some specific examples of such compounds produced by plants?
Secondary metabolites are compounds produced by plants that are important for the survival and propagation of the plants that produce them. Secondary metabolites serve as chemical signals enabling the plant to respond to environmental cues or functioning in the defense of the producer against herbivores, pathogens, or competitors. Other secondary metabolites provide protection from radiation from the sun or assist in pollen and seed dispersal. They are produced at various sites within the cell and stored primarily within the vacuole. The three major classes of secondary plant compounds are alkaloids, terpenoids, and phenolics.
What are aerial roots?
Aerial roots are adventitious roots formed on above-ground structures such as stems. Aerial roots serve different functions in different species. In some species, such as the banyan tree, Ficus bengalensis, and red mangrove, Rhizophora mangle, the aerial roots are called prop roots since they support the plant. The aerial roots of ivy, Heterohelix, ball moss, Tillandsia recurvata, and Spanish moss. Tillandsia usneoides, cling to the surface of an object providing support for the stem. The aerial roots of the vanilla orchid, Vanilla planifolia, and philodendron are photosynthetic.
How do brain cells function differently in people with Alzheimer's disease?
The buildup of toxic proteins, plagues, in the brains of Alzheimer's patients is linked to changes in several genes that regulate memory and learning. The protein responsible for this effect is a small peptide, beta amyloid. This protein is made by the body throughout the entire lifetime. But in Alzheimer's patients, either too much is synthesized or too little is broken down. The buildup of plaque has been implicated in neuron death, which eventually leads to dementia. Researchers have studied mice that were genetically engineered with genes to cause them to accumulate beta amyloid deposits in their brain. The mice developed neurological problems consistent with Alzheimer's disease that occurs in humans. Results from the tests performed on mice indicated that the functions of at least 6 genes were suppressed in the presence of the renegade protein.
Are tortoises and terrapins the same as turtles?
The terms turtle, tortoise, and terrapin are used for various members of the order Testudinas. From the Latin term testudo, meaning tortoise. In North American usage they are all correctly called turtles. The term tortoise is often used for land turtles. In British usage the term tortoise is the inclusive term, and turtle is only applied to aquatic members of the order.
What groups of organisms have a cell wall?
A cell wall is present in organisms in the kingdoms Archaebacteria, Eubacteria, Protista, fungi, and Plantae. Animals are the only organisms that do not have a cell wall.
How can the results of a genetic cross be predicted?
The results of a genetic cross can be predicted using a diagram called a Punnett square, which graphically represents Mendel's laws of segregation and independent assortment. On each side of the square, one places the possible alleles donated by each parent. Dominant alleles are represented by capital letters G in this case, and recessive alleles by lowercase letters, g. Inside the boxes are the allelic combinations, in this case. Each box would contain GG, heterozygous combination.
Who was Nico Tinbergen?
Nico Tinbergen, 1907 to 1988, along with Konrad Lorenz, 1903 to 1989, and Karl von Frisch, 1886 to 1982. Laid the 4 cornerstones of ethological study: causation, development, evolution, and function of behavior. Tinbergen's earliest studies were of the aggressive threat displays of adult male three-spined stickleback fish, Gasterosteus aculeatus. Tinbergen found that by presenting the male with stickleback models, he was able to elicit an aggressive response. He began his career in animal behavior as a child by studying the mating and nesting behavior of sticklebacks in his backyard pond. He attended the University of Leiden, where he did his dissertation research on digger wasps. His thesis was only 36 pages long and was barely accepted by the faculty. In 1936 he met Lorentz at a symposium, and they became lifelong friends and colleagues. During World War II, Tinbergen was imprisoned in a German hostage camp and Lorentz in a Russian camp. When they were reunited after the war, both resumed their studies on animal behavior.
Who is considered the founder of modern conservation?
American naturalist John Muir, 1838 to 1914, is the father of conservation and the founder of the Sierra Club. He fought for the preservation of the Sierra Nevada in California and the creation of Yosemite National Park. He directed most of the Sierra Club's conservation efforts and was a lobbyist for the Antiquities Act, which prohibited the removal or destruction of structures of historic significance from federal lands. Another prominent influence was George Perkins Marsh, 1801 to 1882, a Vermont lawyer and scholar. His book Man and Nature emphasized the mistakes of past civilizations that resulted in destruction of natural resources. As the conservation movement swept through the country in the last 3 decades of the 19th century, a number of prominent citizens joined the efforts to conserve natural resources and to preserve wilderness areas. Writer John Burroughs, 1837 to 1921, forester Gifford Pinchot, 1865 to 1946, botanist Charles Sprague Sargent, 1841 to 1927, and editor Robert Underwood Johnson, 1857 to 1937, were early advocates of conservation.
What are the functions of the central vacuole?
The central vacuole can encompass 80% or more of the cell. It is usually the largest compartment in a mature plant cell, and is surrounded by the tonoplast. The important functions it provides include storage, waste disposal, protection, and growth.